Hi, my name is Amy Sullivan, and I'm the chair of the AAAR Endowment Committee. It's my pleasure to welcome you to our fifth in our monthly series of AS&T lectures. This is a new initiative being supported by the Freelander Memorial Fund. With this idea, we hope to be able to highlight the amazing research happening in our community, tie our journal to other activities, and also give us all an opportunity to get together outside of the annual conference. So each month, the editors of AS&T select a high impact paper to be presented by its author. These lectures are being recorded, and you can access them through AAAR's new YouTube channel, which you can find through AAAR's webpage under the events tab. In addition, each month, one of our student chapters is hosting, and the student chapters have also created a journal club that meets a week before each lecture to discuss the paper to be presented. So students keep an eye out for that email about that. With that, I want to thank everyone who's helped to make all of these lectures possible, and also thank you for joining us. And so now I'll turn it over to our student chapter from the University of California, Riverside. Thanks.
Thanks, Amy. Hi, everyone. My name is Ryan, and I'm a PhD student at UC Riverside, and it's my pleasure today to introduce our speaker, Fobang Liu. Dr. Fobang Liu is a postdoctoral research at the School of Chemical and Biomolecular Engineering at the Georgia Institute of Technology. He earned his doctoral degree at the Max Planck Institute for Chemistry. His current research focuses on understanding the health effects of atmospheric particulate matter, and he has pioneered the application of novel tools and interdisciplinary knowledge in PM toxicology study. He's combined laboratory chamber and ambient field measurements to study the chemistry of PM and developed advanced in vitro and in vivo systems to link PM composition to its toxicity and explore the PM toxicologic mechanisms. These systems include microfluidic single-cell assay, as well as a developmental genetic model system, C. elegans, that expresses a variety of functional biomarkers and gene expression reporters. After the talk, we'll have about 15 minutes for questions at the end. Feel free to ask questions in the chat as they come up, or raise your hands when we get to that, and we'll work through any questions in order. And with that, I'll pass it off to you, Fobang.
Okay, thanks for the introduction, and I hope everybody can hear me. So it's my honor to be here, and I'm very happy to have this chance to present our review article. So in this article, we introduce the recent progress in the development and applications of microfluidic techniques for in vitro toxicity studies of MSPM exposure. This is because we see more and more relevant studies are coming out, and we see the great potentials of the microfluidic techniques in overcoming some limitations in the traditional formats. So we think it's good to share what we know to our Aerosol community. And yes, this is a review article, and we have discussed a few research papers that using microfluidics for different measurements or assays. So I will first briefly introduce the assays that will be mentioned in this presentation, and then I will talk about how the applications of microfluidic techniques in both chemical and cellular assays can overcome some limitations in the traditional formats. So there will not be a lot of scientific questions, discussions inside, but it's more like an overview of what the advantages of each microfluidic platform have compared to the traditional formats, and what can be further improved. And finally, is some thoughts for the future directions and challenges. And so I believe in our Aerosol community, we all know ambient PM is bad to our human body and to our health. And so we know millions of deaths per year could be attributed to PM exposure. And the PM is the only environmental risk factor among the top 10 for premature death. And also some studies have revealed that there's no clear threshold of PM 2.5 mass concentration for a safe exposure. And regarding the impact of PM 2.5 on human body, so it's well recognized that the impact is beyond the non-system, so it can reach to the heart, the liver, and the brain. And accordingly, so PM 2.5 exposure has been associated with a broad range of diseases. And there's also a concession that only using the PM mass concentration is not enough to understand the PM toxicity, because PM is a mixture of hundreds of compounds, and it has the characteristic of shows the special temporal variation. There are many PM properties may affect its toxicity, like the size, the chemical compositions, and other factors listed here, or even not listed here. And on the other hand, PM may cause a number of possible biological effects. So what could be used as a good indicator to evaluate the PM toxicity, and what are the biological mechanisms in causing these effects or diseases? And more important, what are the underlying connections between the PM properties and the biological effects? So there are a lot of scientific questions inside. But besides that, we also need to establish or apply powerful approaches that can meet the different requirements for the corresponding scientific questions. So currently, there are both in vitro and in vivo models that have been used to study the PM toxicity, and each experimental model has its advantages and limitations. So the in vitro model, including chemical assays and in vitro cellular assays, these are the common approaches to evaluate the PM toxicity. Also, link the different health endpoint measurement to PM properties. And in vivo models, including some small model organisms and the mouse model, which is the most used in vivo model. So these in vivo models have a higher homology with humans, and they can be used to study the systemic effects and multiple tissue effects. And if for some drug developments to prevent the PM health effects, so the animals that have higher homology with humans or even humans need to be involved. And today my focus is on the in vitro assay methods, that is the chemical and cellular assays. So these in vitro assays, they are cost effective and easy to perform. Also, they're faster than the in vivo models because they have a shorter life cycle. And these advantages make them easier to be applied to analyze the different endpoints from a large number of PM and link the PM exposure to the different measure response. And so today my focus is on the chemical assay and cellular assay, also the microfluidics, and I also give a brief introduction on each of them. So the chemical assay is used to measure the oxidative potential of PM. The oxidative potential describes the capacity of the PM in characterizing ROS generation. So this because the oxidative stress, which could result from the overproduction of ROS, is a potential mechanism of the PM toxicity. And usually a depletion of chemical process is measured to represent the OP of the PM. And these chemical processes are mostly antioxidants like the DDT and ascorbic acid. And there are some online automated systems have been developed for OP assays. So, and these online systems can save some labor work, and they can provide high temporal resolution of the OP measurement. For the cellular assays, the general protocol usually takes a longer time and is more labor intensive than the chemical assays. This is because on one hand, the cell culture takes time for equilibration. Also, the cell response may not be instant when exposed to PM. However, with the cellular assays, we can not only use it to evaluate the toxicity of different types of PM, but also with the cellular assays, we can investigate the signal transduction in response to PM, and this is not possible by chemical assays. And for the microfluidic development, so it was first emerged as a microelectromechanical systems in the 1960s. And after George Whiteside introduced the rapid prototyping and replication of some polymers like PDMS, so the microfluidic technology has become a fast-growing technology in the past decades. And today, there are a lot of materials can be used to fabricate microfluidic device as an alternative to silicon processing. And the microfluidics have demonstrated their benefits in the application, such as the joint microfluidics and organ-on-chip. So the general advantages of the microfluidics include low consumption of reagents, and it's flexible of device design, and also the integration and automation of assays. So microfluidics have been applied to diverse research from biology, medicine, to chemistry, and also to MSc Aerosol Science. And there are two recent review articles summarizing the applications. One is more on the general aerosol science, and the other is more specific to bioaerosol sampling and detection. And today I will be presenting the application of microfluidics in aerosol toxicology. And first is the applications of microfluidics in chemical assays. The first one is about development of an online OP monitoring system. So we know mostly current the OP assays are offline analysis with PN connection and extraction step. So there are some limitations in such protocols. One is the PN connection takes a certain time from several hours to several days. And second, during the PN connection or storage and extraction, some unstable or reactive components, you may lose this information. And there's some online systems have been developed to overcome these problems. And these online systems usually involve a direct PN sampler and coupled to an automated analytical system. And in the system developed by Semler et al. In 2012, they use a microfluidic electrochemical sensor and coupled to a pulse. And the microfluidic electrochemical sensor can directly analyze the remaining DDT after the DDT reacting with the PN, because the sensor is very sensitive and selected to the fire groups in DDT. And this eliminates the need for quenching and developing reagents in the traditional DDT assays. And so they further demonstrated that online system can provide a higher temporal resolution compared to other online systems. It's because with the microfluidic electrode sensor, only a small amount of PN mass is needed, and so the PN connection can be very short, and they reported a high temporal resolution as of 3 minutes. And the required PN mass is around 3 orders of magnitude lower than in the traditional method. And in one application of this online system, they use it to monitor the OP from lab-generated aerosols, and they observe a better correlation between the DDT consumption rate and PN mass compared to the traditional method. So it's a very promising platform, but to our knowledge, we haven't seen the application of this platform for field studies. So we don't know how's the stability and the maintenance of this microfluidic sensor for the long-term measurement, and this should be further evaluated. And the second application of microfluidics in chemical assay is about the employment of paper-based analytical device for fast screening of OP. So this micropad device is first developed for point-of-care medical diagnostic. And recently, it has been extended to the analysis of food safety and also for the environmental analysis. And one common method for fabrication is the wet printing. So in this process, only a wet printer and a heater are needed, and the process is very fast, inexpensive, and no need of any solvents. So the micropad device have been applied to two OP assays. One is for the DDT assay. So the procedures to perform a DDT measurement with the micropad is very simple. All you need to punch a portion of filter and then add DDT solution on top of the filter and then place the filter on the micropad and then use some aqueous solution to elute the remaining DDT to the detection zone. So there's some advantages of this micropad device. First, it doesn't need PN filter extraction, and also it reduces the PM mass, and it's portable, and the cost is very low for each device. So these advantages make it very easily to apply for a personal exposure assessment when combining with a PN sampler. So and indeed, the office, they have asked some volunteers to carry out both the PN sampler and the micropad device and validate that micropad device are capable for personal exposure at 3 different events. And the second application is of the micropad is on the glutathione assay, is another antioxidant for OP measurements. So the design of this device, the difference compared to the previous one is that all the steps in this protocol can be done on chip. And usually they use a scanner and image processing software either to analyze the color intensity or by a distance-based detection. So these detection methods are easy to perform, but they're always semi-quantitative and with a low sensitivity. So further improvements can be apply some high-sensitivity detection strategy, such as fabrication of electrochemical paper-based analytical device, as this picture shows here. And another thing can be further improved is that so the wet-based flow channels are only amenable to aqueous flow, and so alternatives that are compatible with organic solvents should be incorporated, and so the water insoluble PN components can also be accounted for the OP measurements. Based on the advantage of the micropad systems, they are compatible, complementary to link the OP results with the health outcomes from epi studies at the personal level. We know right now that for the conventional OP assays, we connect PN at a fixed sampling site or location, and the OP results from these measurements are usually considered as a population level exposure assessment, and this micropad systems can be an alternative for the personal exposure assessment. And then I will move to talk about the microfluidics application in several assays. And the first one is about developing microfluidic platforms for high throughput and high content screening of multiple endpoints. So we know the cellular signaling pathways usually involve a number of macromolecules such as proteins, but the immunoassays for protein detection usually include a few steps, and the conventional protocols are labor intensive. There are a lot of pipetting steps involved, and it's time consuming. Also, more important is it requires a large volume of samples and reagents. And these limitations are exactly what the microfluidics can overcome. For example, in a study by Chen et al. In 2017, they developed an on-chip protein microarray assay. So in this microfluidic system, they have a concentration gradient generator, a cell culture chamber, and an immunodetection chip. So this system enables the PN dilution, cell culture, as well as the immunodetection. All can be done on chip. And importantly, so multiple proteins can be analyzed simultaneously on chip. And it only requires a minimum amount of PMS. So it's also around 3 orders of magnitude lower than in the traditional method. And right now, the concentration gradient generator they use is a linear dilution, where we know the dose-dependent cellular response to the PN exposure or to the drug is usually behave as a logarithmic manner. So future studies should use a gradient generator that can span several orders of magnitude. And the next application of microfluidics in cell assays are development of microfluidic single cell assay to study cell heterogeneity. So cell heterogeneity is an inherent property. That means individual cells that may show difference on the gene expression and also on the response to external stimulants and so on. But currently most assays are bulk assays. That means the measured results are the average from hundreds or thousands of cells. And assuming all these cells, they have similar behaviors. But as two examples show in these two figures, so the conclusions could be biased by only using bulk data. For example, if the cells that are not or having the same similar response as the bulk average, like if it is a bimodal distribution, and the population has with a higher response, they may have a different downstream outcomes, and this cannot be reflected by only using a bulk measurement. And the flow cytometry is often the choice of technology for single cell analysis, but it is not capable to monitor temperature change and provide special information, for example, some subcellular information. So we need to develop platforms that can confine cells in a small space. And the microfluidics have been developed for such purpose. So one example is from our group. So in this study, we developed a microfluidic single cell check array that we can confine cells in a small chamber. And so the cell identity can be maintained over time. And as a proof of principle study, we investigate the cell-to-cell variation in RS response to ambient PN extract. And with this device, we can check the temporal behavior of single cell response. And by obtaining this single cell information, we can do more in-depth analysis and provide more detailed information for the PN toxicity evaluation. For example, we can summarize the cumulative fraction of response cells over different time points. And we also can perform some statistical analysis to understand the underlying behavior patterns around these single cells. Another interesting application using the microfluidic single cell check array is to monitor the nanoparticle uptake. For example, in the study done by Curia Metals et al. In 2016, and they isolated the single cells and then exposed to 3 different nanoparticle concentrations. So And it's a video, but it's not working, sorry. So they isolated single cell to 3 different nanoparticle concentrations. And the result revealed that there's a heterogeneity in time and amplitude of cell response to similar stimulus. And in addition to these 2 applications I mentioned here, there are also other microfluidic-based single cell systems have great potential in evaluating the PM toxicity at both cellular and molecular levels, although they haven't been applied to the PM toxicity study. So some representative technology are the single cell omics studies, and also some single cell immunoassays. Okay. So the first application of microfluidic in cellular assay is the nanochip model to mimic physiological relevant conditions. So we know the current cell assays usually is a mono cell culture with a single cell type. And this system is not adequate to assess the organ toxicity because it excludes the interactions with the labeling cells, and also exclude the interactions with extracellular metrics. And some studies already show that the cold culture systems show a different sensitivity to the external stimulus compared to monoculture systems. And here they also show that the on-chip monoculture is more sensitive to off-chip monoculture. And since the cold culture systems, they improve the simulation of in vivo situation, and they can be used to study the translocation of nanoparticles from PM exposure. So they can be a variable tool for the PM toxicity studies. And one early and representative study is from Hu et al. In 2012. So in this study, they reconstruct the nanochip model by a 3D cell culture. So they include both epithelial and endothelial cells, and in between is a permeable membrane. And there's air introduced in the epithelial channel to mimic the air-liquid interface within the nun. And besides that, they also include 2 side chambers to mimic the mechanical strain by breathing movements. And with this nanochip model, they have seen some very interesting results. So they exposed the 100 microgram per mil silicon nanoparticles to this nanochip model and measure the RS generation. So if there's only nanoparticles, or if there's only 10% mechanical strain, they don't see any RS generation within the first 2 hours. But if there's nanoparticles with 10% mechanical strains, they see high levels of RS generation within the 2 hours. So they think the mechanical strain might act as a surgeon with nanoparticles for RS generation. And they also use this model to study the nanoparticle translocation. And they also see when there's a 10% strain applied to this nanochip model, the translocation rate can increase a lot. And later, when adding an antioxidant to this model, they see a decrease of the translocation rate. So this result suggests that the elevated RS generation by the mechanical strain might be responsible for the increase in the barrier permeability to nanoparticles. So these results cannot be done by only using a monocell culture system. I will also say, and it's not easy to be done by using animal models. So the nanochip model can be a variable alternative to study the complex cellular response. And recently, some researchers and even proposed the human body on chip concept, because there are different organs on chip already have been developed, and we know exposed to PN has been linked to diverse systemic effects. So they propose to integrate multiple organ chip platforms to create complex interactions between the human organs. And this will facilitate visual studies of systemic effect caused by PN. And this human body on chip model can serve as a low-cost alternative to animal studies. Okay, that's all I want to introduce. So in summary, we have seen the microfluidic techniques have shown the versatility to overcome many of the limitations in the traditional chemical and cell assays. And for chemical assays, so it has been derived from an online monitoring system, which can provide high temporal resolution results. And also, the paper-based device has been employed for the organ measurement, and it is suitable for personal exposure assessment. And this is especially useful for a cohort study. And for cellular assays, so there are different microfluidic platforms have been developed. So the microarray assay are able to analyze multiple endpoints simultaneously. And for also some single cell check array that can study cellular response at single cell level. And language model can be used to study organ-specific effects. And looking forward, the application of microfluidics in aerosol toxicology is still at an early stage. So there's still a lot of potential applications of microfluidics in helping the aerosol toxicology study. For example, some microfluidic-based systems in drug screening and nanomaterial toxicology, they are promising to be applied to the aerosol toxicology. Also, we should develop some microfluidic platforms with an adequate interface exposure to PM, which can preserve the physical and chemical properties of the PM before exposing to the cells. And also some of the ones, organs or human brain organoid on chip models should be applied to study the aerosol toxicology because the PM can cause systemic effects. But besides that, efforts should also made to establish standardized platforms and protocols in order to facilitate the comparisons among different systems. And also efforts should be put forward to the production of end user device and scalable products, because at the moment, the fabrication of micro platforms still need a lot of expert operator and a large set of specific equipments. And that's all my presentation. And finally, I want to thank my supervisors, Dr. Sally and Dr. Hanu, and all the members in LULAB and INEP. And it's been a lot of fun working in two courses with completely different research. And with that, I would like to thank you for your attention.
Thank you for a great talk, Fobang. I see a virtual round of applause coming in there. We do have a bit of time for questions here. So if you want to either unmute, put those in the chat, or raise your hand, we can start working through questions as they come up.
So is there a big difference between the immobilized platforms and the droplet-based mobile platforms for the cell-based analysis? Does either one of them have a significant advantage?
Sorry, so your question is the advantage of immobilized platform and some mobile platform like Jobic microfluidics?
Sure, yeah. So Fluidigm, as an example, versus one of the ones that uses droplets instead to, like David Weitz's droplet platform as an example?
Yeah, so for the immobile platforms like the single cell array that we had and developed, so we can have the temporal behavior response, right? But with the Jolly platform, so we cannot keep the cells in for such a long time. But on the other hand, the Jolly platforms, I think there's some also Jolly. So with the Jolly platforms, we can do more other alternative measurements like the Jolly single cell RNA sequence and all these molecular informations.
And are any of those assays just live dead types of assays?
Live dead type assays?
Yeah, so for the cells, so exposure to various different concentrations and then either vitality or live dead types of assays.
You mean by using the microfluidic techniques?
Yes.
Yes, I think there's some also. I don't see any platforms that are specific to investigate the live dead cell assay. But usually, when we establish or develop a micro platform for cell culture or other purpose, like for our single cell RS response, before that we also need to do a live dead cell assay to ensure that the cells they perform well or perform normal in the microfluidic device.
Sure. Thanks.
Thanks.
Let's see, we'll give it another minute. I have one in the meantime. I was wondering if you could talk more about the cellular responses from on-chip culture being more sensitive to the off-chip culture that you mentioned, and if there'd be a similar contrast to the on-chip culture versus the natural biologic environment in that case.
Yeah, so yeah, I'm not surprised to seeing the on-chip is more sensitive to the off-chip. One hand, because usually there's still a flow coming to the cells for the on-chip culture. On the other hand, like the shear stress or the microenvironment is more. So the shear stress is higher for the on-chip than the off-chip, right? The off-chip is usually in static condition.
All right, thank you.
Maybe I can ask another question as well. For each of the studies that you referenced in this paper, I wonder if there is significant variation in the particulate matter source that you use, maybe in terms of chemical composition or just the source, outdoor, indoor, and would this affect the outcome that you see for if the microfluidic techniques outperform the traditional assays?
Which application you mentioned?
Just for any of them, but maybe for the second one, for example, the second application.
The second means the single cell one, or?
Yes, yeah.
So for this one, we haven't analyzed the different PN samples with distinct PN compositions. So as I mentioned, this is only a proof of principle study, and we only use one PN sample, but with different concentrations. So I'm not sure if when we see and when we use PN with different composition, what the result could be.
Okay, great, thank you.
Go one more pass if any more questions pop up. And I see a question in the chat. I'll read it off. Do you know if anyone has tried making aerosol microfluidics for extreme environments like high temperature exhaust?
No, I haven't seen. Yeah.
All right, if we don't have any more questions, we'll give you one more thank you and wrap up here. To add to the scheduling, next month's seminar is currently scheduled August 19th. And we do have one more question snuck in.
Okay.
If you can pull up slide 13, how do you explain the apparently, I'll wait for you to pull it up actually, it might be easier to go through with the visual.
Slide 13.
Yeah, 13.
And what's the question?
How do you explain the apparently antagonistic effect of PM 2.5 in the PM 10 consumption of DTT?
The difference between the PM 2.5 and PM 10?
I believe so. I'm open to corrections if I'm reading that question at a different interpretation.
So if I remember, I'm not sure if they discuss much on the results between the PM 2.5 and PM 10 in their paper. So the goal of this figure is to show that they have applied the device for some personal exposure assessment, like some people in the clean day outdoor and some people in different events like smoke day and kitchen day. But yeah, I don't know how to interpret the difference between the PM 2.1 and 10.
All right, well, if we're all covered for questions, then we may wrap up here. Thank you again, Dr. Liu, and we'll see everybody for the next seminar next month on August 19th.